we can start. So, good afternoon, good morning, everybody, and welcome to this liver seminar Young Investigators edition. And first of all, I would like to thank Liver the opportunity for our group to host this seminar today, and also thank my group PI, Dr. Ruben Frances, for offering me also this chance to host our speaker. So it's my great pleasure to introduce you to our speaker today, Dr. Cristina Llorente. Dr. Llorente is assistant professor in the Department of Medicine in the School of Medicine at the University of California, San Diego, and member of Professor Snavels Laboratory. She graduated from Autonomous University of Madrid with a PhD in biochemistry, molecular biology, and biomedicine. And while there, she clarified the role of COX-2-dependent prostaglandins in chronic liver disease, hepatitis, fibrosis, and hepatocarcinogenesis in the laboratory of Dr. Paloma Martin-Sanz. After her doctorate, she completed a fellowship at Dr. Gerald Solewski's laboratory at UC San Diego, studying the pathogenesis of diabetes and obesity. As a postdoctoral fellow in Professor Snavels' laboratory, she has focused in understanding the influence of microbiota in liver diseases and metabolic disease. Later on, she studied the role of Enterococcus in alcohol-related liver disease, its potential as a marker of disease, as well as the possible development of preclinical therapies to counteract the illness. In recent times, Dr. Llorente is exploring whether the modulation of small intestinal globule cells acting as sentinels of the intestinal homeostasis is a good strategy to prevent translocation of bacteria and to reduce alcohol-associated liver disease. Her long-term goal is to build a research program dedicated to mechanistically understand the role of host microbiota interactions in modulating inflammation, the immune system, intestinal homeostasis, to decipher the cellular and molecular mechanisms involved in the progression of liver diseases, and to discover therapies and molecular targets to alleviate it. Cristina, thank you for accepting our invitation today, and it's your turn now.
Hello, everybody. Thank you for joining here today, this Liver Seminar Young edition. And thank you, Esther Caparrós and Dr. Frances, for inviting me here today to give this presentation. Thank you for this long introduction. So I would like to thank the organizers, and also I would like to also acknowledge the sponsors for allowing and supporting this event. And as Esther has mentioned, during my past years, I've been interested in understanding the role of host microbiome interactions in liver diseases, and I personally focus more in alcohol-associated liver disease. Alcohol-associated liver disease represents a spectrum of liver injury that is produced by ethanol. This spectrum of liver injuries includes alcohol hepatitis, includes steatosis, includes steatohepatitis, and fibrosis, and more advanced forms of liver injuries such as alcohol hepatitis, which produce a mortality of 20 to 40% at 1 to 5 months. As well as alcohol-associated cirrhosis, and also acute alcohol hepatitis, here represented as an acute on chronic liver damage that has an estimated mortality of 75%. Alcohol-associated cirrhosis is also a factor to develop hepatocellular carcinoma. Alcohol-associated disease is a major cause of liver disease worldwide and liver transplantation in Europe and the States. And incidence and mortalities of alcohol cirrhosis and alcohol hepatitis are rising. They occupied, alcohol abuse occupies the 7th position of the risk of death. And the treatment for these diseases reminds us as it was 4 decades ago, which is abstinence, nutritional support, and the use of corticosteroids, which are known to prompt infections. The need of a therapy is imperative. And early transplantation is the only curative therapy that is offered right now only at selected centers and only to some patients. Until recently, the human microbiota was largely a mysterious black box. Cultivation in vitro was the cornerstone of microbiology since the 19th century. However, now DNA-based technologies have expanded our horizons, providing new data sets, providing more meaning and biological sense to the human microbiota and their collective genes, the metagenome. It is estimated that the total number of bacteria it is in the same order of magnitude as the number of human cells. And the microbiota genome, it is providing 360 times more genes than the human genome. This gives us an significance of the relevance of our friendly symbionts. This principal component, blood, is showing us how the microbiota is distributed according to different body areas. All of the environments that are exposed, all of the body areas that are exposed to the environment are colonized by microbes. In this other graph, we can show the distribution of the distribution of the microbial colonization is dependent to each individual and according to different body surfaces. The metagenomic carriage of metabolic pathways is more stable among individuals. Microbiome alterations are associated with a vast plethora of diseases, among them alcohol-related liver disease. And ethanol abuse is associated with dysbiosis and also with bacterial overgrowth, as early as 3 weeks in, especially and more pronounced in the proximal small intestine. Patients with alcohol liver disease are known to have reduced levels of bifidobacterium and lactobacillus, Faecalibacterium prausini, Rominococcus, and it has an increase in lactose species and also reduction of bacteroidetes. In 1984, Bode et al. Already demonstrated an increase in bacterial overgrowth in samples, in jejunal samples from alcoholics. Schnabel group also showed that alcoholic patients in the duodenum had an increase in mucosal associated bacteria. Indeed, microbiota alterations leads to intestinal barrier dysfunction. This intestinal disruption is going to promote the translocation of bacteria to mesenteric lymph nodes and to the liver, increase translocation of which is going to promote the progression of liver diseases. Increased translocation of the bacteria is going to promote and activation of the lamina propria immune system, which is going to induce the secretion of inflammatory mediators, which is going to amplify this barrier dysfunction. Besides alcohol and other microbial products are also promoting the inflammation in the liver and also hepatocyte deaths and promotion of fibrosis. Related to this, there is right now a clinical trial, human clinical trial, that is using hyperimmune bovine colostrum and is showing promising results. It's showing promising results. It's been shown to reduce the translocation of bacteria. In addition, also a compromised ethanol immune system in the liver is also going to exacerbate this condition. Certainly, alcohol-related liver disease can be transmitted via fecal microbiota transplantation. Humanized gene-free mice and conventional mice were transplanted with intestinal microbiota from patients with and without alcoholic hepatitis. These mice fed with a controlled diet, and especially those fed with alcohol diet and transplanted with intestinal microbiota from severe alcoholic hepatitis patients had an increase in severity of liver disease demonstrated here by ALT. Understanding the molecular mechanisms of altering the microbiota could be a good potential alternative therapies to prevent the progression of liver diseases. The underlying mechanisms of dysbiosis and bacterial overgrowth is not well characterized. However, it's thought to be related to intestinal changes in intestinal dysmotility, changes in the gastric pH and the gastric acid, and also changes in the innate immune system. The gut microbiota regulates the intestinal immune system and is crucial to maintain the perfect balance in the intestine and a healthy condition known as intestinal homeostasis. Gene-free mice and conventional mice here were subjected to an acute liver alcohol model, and that is mimicking binge in humans. Gene-free mice had increased levels of ALT and also increased steatosis, among other parameters, indicating that microbiota is essential to prevent liver injury after ethanol abuse. It's important to mention that gene-free mice had significant deficiencies in the immune system and have lower levels of IgA, T regulatory cells, and reduction of antimicrobial peptides, among other deficiencies of the immune system. Taking the relevance of the microbiome and the onset of alcohol-associated liver disease into account, manipulation of this environment should be considered. The story of the use of the story of the success of the use of antibiotics has led to conflicting results and different viewpoints. It's been extensively shown that antibiotics in preclinical models, in rodent models, are effective to combat liver disease. However, for example, in this clinical trial, using a non-absorbable antibiotic known as paromomycin, treating 50 patients with alcohol-associated liver disease, among them 27 with cirrhosis and 33 with cirrhosis, and had no changes in the endotoxin concentrations or liver function test. However, the use of more personalized, one of the reasons why what could be the reason that this antibiotics therapy did not work? It is maybe possible that antibiotics are affecting the immune system, and also maybe a more personalized medicine or antibiotic treatment dependent on the type of dysbiosis of the patients could be more effective. However, the use of antibiotic as a prophylactic in patients has been shown to be effective. For example, in these clinical trials with 100 patients with cirrhosis and low protein concentration in aseptic fluid, receiving ciprofloxacin in 50 patients and 50 patients without it, they had reduced risk of infections and improved survival. As I mentioned, alcohol-associated liver disease is a transmissible disease. I previously mentioned that we can transfer severity of liver disease severity. But now, can we prevent it by transplanting with donors, healthy donors? This in animal models, transplanted with fecal microbiota transplants from resistant donor mice to alcohol-sensitive recipients confer resistant to the disease. Fecal microbiota transplantation to severe alcohol hepatitis patients has shown to improve to improve severity in these two clinical trials with this amount of patients. Here, they had a survival of 87.5%, and in this other clinical trial, they had a survival of 75% after the use of fecal microbiota transplantations. There is right now a clinical trial going on in cirrhotic patients and alcohol use disorder patients to assess the effectiveness of fecal microbiota transplantations. Fecal microbiota transplantations to patients with cirrhosis with hepatic encephalopathy has also demonstrated to improve cognition, reduce hospitalizations, and lower the MER score in these two clinical trials. More studies using fecal microbiota transplantation to treat patients with cirrhosis and hepatic encephalopathy has also shown to improve duodenal mucosal diversity, dysbiosis, and reduce inflammation, as shown here, increase in antimicrobial peptide expression, and reduce the expression of LPS binding protein and improve cognitive performance. In this case, they used 10 patients and they gave them these oral capsules, and they give them 15 capsules at a time. And then in this other, with the same clinical setup, they also reduce inflammation, reduce LVP, improve cognitive performance, and interestingly, they also found increased the conjugation to secondary bile acids. In addition to fecal microbiota transplantation, other therapies such as probiotics has been shown to be beneficial. And the use of probiotic in preclinical studies has also shown improvement of gut microbiome homeostasis and reducing liver damage. So in these clinical trials, using supplementation with Bifidobacterium bifidum and Lactobacillus plantarum to alcoholic hepatitis patients, they showed to reduce severity of the disease after probiotic therapy. In this other clinical trial, using Lactobacillus subtilis and Streptococcus faecium supplementation to alcoholic hepatitis patients, also they show an improvement after the use of probiotics. There is right now another clinical trial in phase 2 that is being done in alcoholic hepatitis patients and using Lactobacillus rhamnosus GG to show the efficiency of the use of probiotics. Other therapies such as prebiotics and nutrition support such as long-chain fatty acids, medium and long-chain fatty acids, as well as therapies related with FXR manipulation are also being tested. Knowing the relevance of microbiota in the onset of alcohol-related liver disease, how does factors that influence the microbiota affect alcohol-related liver disease? 36% of patients with alcohol-dependent patients and as many as 67 to 72% of patients with cirrhosis are using acid gastric medication. Among these drugs are proton pumps inhibitors, which are known to promote dysbiosis. And knowing these facts, we hypothesize whether proton pumps inhibitors, PPIs, are related to alcohol-related liver disease. To answer this question, we use a model of liver decay for 9 weeks, and then we expose these mice that are a genetic mouse model that has a point mutation in the proton pump 84 that is important for the secretion of hydrochloric acid to the stomach. Therefore, this mice has a gastric pH of 7 when the normal mice have a pH of 3 in the stomach. We found that impairment of gastric acid secretion in the mutant mice for ATP4 had an exacerbated ethanol-induced liver disease. We observed that levels of ALT and were significantly higher, as well as the steatosis indicated by higher levels of triglycerides, as well as they had also increased collagen deposition and increased inflammation, here represented as an increase in the levels of IL-1 beta. Next, we studied the composition of the microbiota by doing 16S RNA sequencing analysis, and we found that the composition in these different animals are different among the different groups and the different treatments. Notably, absence of the acidic environment promotes an expansion of the numbers of Enterococcus in the fecal samples. And also, this Enterococcus was able to translocate to the mesenteric lymph nodes and to the liver. To confirm previous results, we used a pharmacological approach using 200 ppm of omeprazole and also 9 weeks of flavored Again, we found increased levels of ALT and increased levels of steatosis, increased collagen deposition, and inflammation. And these results support the hypothesis that impairment of acetic acid is related to exacerbation and progression of ethanol-induced liver disease in mice. Also, when analyzing the microbiome in this pharmacological model, we found an expansion of Enterococcus that was able to translocate to the mesenteric lymph nodes and the liver consistently. To investigate whether Enterococcus is involved in the exacerbation of the disease and the causative role of the previous models, we established a model of colonization of bacteria. And according to the 16S, the most abundant species of Enterococcus was Enterococcus faecalis. And we needed to establish a model of colonization where the number of Enterococcus was similar or lower, but never which needed to avoid to gavage to the mice and that the amount of Enterococcus was higher in intestine. So we achieve these numbers because Enterococcus faecalis, when we gavage, the Enterococcus faecalis was lower than the other previous models. After gavaging E. faecalis and exposing these animals for to 9 weeks of liver decay, we found an increase in liver disease, not only after ethanol feeding, but also in the using control diet. We found increased levels of ALT and also increased steatosis. After gavaging E. faecalis, we found increased collagen deposition and also more inflammation. We then examined the association between the PPI use and the development of alcohol-associated liver disease. And for that, we used 4,830 chronic alcohol abusers. These three groups consisted in 1,000 active users of PPIs, 745 previous users, and 3,000 never users of PPIs. The 10 cumulative risk of alcohol-associated liver disease was 20% in the active users and 16% of in the previous users and 12% in the never users. In addition, we found also an increase of Enterococcus in alcoholic patients and also in healthy humans not represented here. They also had an increase and expansion of Enterococcus, consistent with the mouse model. So Enterococcus faecalis is a gram-positive bacteria that is a commensal. And with the advent of the antibiotic therapy, it has become a multi-drug resistant hospital-acquired pathogen that is highly virulent strains of Enterococcus faecalis are expressed a pore-forming toxin that is called cytolysine. What are the characteristics of cytolysine? Cytolysine, activation of cytolysine operon leads to the synthesis of two post-transcriptional modified peptides, SIL-L and SIL-S and SIL-L, that are going to form the port. This cytolysine can form the port and lyses the cells. So we try to study this specific strain that we used in the previous model that I just presented, and we found that this Enterococcus faecalis strain presented the genes of the cytolysine operon. This is by genomic DNA extraction, and this is after RNA extraction. And interestingly, you can see in this in this blood agar plate that Enterococcus faecalis is able to lyse erythrocytes, and here we have a strain positive for cytolysine, and here we have a negative strain for cytolysine that we will use later. So when we isolated and cultured primary hepatocytes from mice after acute Plasmin-Levien-de-Carli model, we found that the addition of the two peptides was able to promote death in the hepatocytes. As I mentioned before, alcohol-associated liver disease is a transmissible disease, and that can be a fecal microbiota transplantation, and that alcohol hepatitis is a life-threatening and severe condition. We perform cysteine RNA sequencing analysis to determine whether the composition of microbiota in alcohol use disorder patients and alcohol hepatitis patients have altered composition. And we found that by principal component analysis and by Shannon diversity analysis, among others, that the composition of the microbiota is very distinctive, or is very different in these patients. Fecal samples from patients with alcoholic hepatitis had about 2,700-fold more Enterococcus faecalis than controls, as measured by qPCR. Interestingly, 30% of patients with alcohol hepatitis were positive for both subunits of cytolysine, and none of the controls presented cytolysine. Only one of the patients with an alcohol use disorder were positive for cytolysine. Most importantly, 89% of cytolysine-positive patients with alcoholic hepatitis died within 180 days of admission, compared to only 3.8% of cytolysine-negative patients. In the multivariable cause regression analysis, detection of cytolysine was associated with 180 days mortality, even after we adjusted for geographical origin, antibiotic treatments, and creatine, bilirubin, INR, and other components of the MELD score. To determine whether cytolysine contributes to damage mediated by E. faecalis, we gavage conventional mice with a cytolytic E. faecalis or a non-cytolytic E. faecalis. The mice were then placed on a chronic binge ethanol diet. Mice fed with an ethanol diet after they were gavaged with the cytolytic E. faecalis developed more severe liver injury, as indicated by higher levels of ALT, increased levels of triglycerides, and also they had here represented by already staining, and they also had more inflammation, more collagen deposition, and mice had also shorter survival times. We also find that cytolysine we were able to find cytolysine expression in the liver, or cytolysine in the liver of these mice gavaged with cytolytic E. faecalis. We also could detect E. faecalis in the mice gavaged with cytolytic negative E. faecalis here, but obviously translocation of E. faecalis is mediated by the alcohol itself. However, we only detected cytolysine in these mice gavaged with cytolytic E. faecalis. To extend our findings to humans, we colonized gene-free mice with feces from cytolytic positive patients and cytolytic negative patients with alcoholic hepatitis. And consistent with our findings, notobiotic mice colonized with fecal samples from two cytolytic positive patients developed more severe ethanol liver injury indicated by levels of ALT, steatosis, represented by triglycerides, and also they have more inflammation and reduced survival, and also we found the cytolysine in the liver. To further demonstrate the causative role of cytolytic E. faecalis for the development of ethanol-induced steatohepatitis, we investigated the effects of treatments with bacteriophages. Phages are ubiquitous in bacteria-rich environments, including the gut. E. faecalis phages that are highly strain-specific can be isolated, which makes the direct editing of the microbiota feasible. To develop a therapeutic approach to precisely edit the microbiota, we cultured cytolytic E. faecalis from the fecal samples from the patients with alcoholic hepatitis, and then we isolated lytic phages against these specific cytolytic E. faecalis strains. These phages had a cephalophage and myophage morphology, as indicated by transmission electron microscopy image. Notobiotic mice were colonized with feces from two cytolysine positive patients with alcoholic hepatitis and given 3 or 4 different but specific lytic phages against E. faecalis. The phages against cytolytic E. faecalis abolish ethanol-induced liver disease and steatosis, as shown here by reducing the levels of ALT and lowering the steatosis, and also lowering inflammation, collagen deposition, and improving also survival times. And they also had reduction of cytolysine for both subunits and also in the liver. In conclusion, this study demonstrates that alcohol hepatitis patients that present cytolytic E. faecalis have worse prognosis and increased mortality, and a precise bacteriophage therapy treatment will be an alternative and a very good potential therapeutic alternative. I have just presented the work that I've been working previous years, and right now I'm going to introduce my current work and my future objectives. We know that the significance and intricacy of the intestinal immune system cannot be underscored, and that specialized cells within the intestine working together with the immune system are necessary for health. Among these specialized cells that are also working together with the lamina propria to regulate intestinal homeostasis, I became more interested in goblet cells. Renewal is mediated by intestinal stem cells. In the intestine, renewal is mediated by intestinal stem cells located in the bottom of the crypts, and generating precursor cells that differentiate along both the absorptive and secretory lineages. And these cells are known, as I mentioned before, are known to communicate with the lamina propria intestinal and lamina propria immune system, as well with the microbiome, and regulate homeostasis. Therefore, the role of goblet cells are very important for inducing homeostasis, regulated also the immune system, as I will explain later, and it could be a potential also target, could influence liver disease. So what is a goblet cell, and what happened to goblet cells during alcohol abuse? So goblet cells secrete an important component known as mucin that is going to generate the mucus layer, which is an important physical barrier that is going to prevent the translocation of microbes and protects against luminal pathogens. MAC2 is the major secreted component of the mucus layer throughout the whole intestine. And while mucin can keep most of the pathogens from penetrating, it is also important because it's going to provide a physical structure to maintain to maintain and content important components such as immunoglobulins and antimicrobial peptides. And also goblet cells are known to secrete antimicrobial peptides. However, mucin serves also as a nutrient source to commensal bacteria and potential pathogens. And it is important to consider that the thicker that the mucous layer is, the more diluted antimicrobial peptides are. A Schnabel group showed that patients with alcohol use disorder had an increase in the thickness of the mucosal layer in duodenal biopsies as compared to non-alcoholics. Now, in collaboration with Peter Starkel and Luca Macchioni from UC Louvain in Belgium, we have now demonstrated that patients with alcohol use disorder have significantly higher numbers of goblet cells in the duodenum. It's been reported by a Schnabel group that a deficiency in MAC2 in mice protects from ethanol-induced liver injury in an intragastric hybrid feeding model of continuous ethanol infusion in mice. Again, mucin is a source of energy for bacteria, and in fact, MAC2 deficient mice had a reduced amount of intestinal bacteria. The mucosal immune system should efficiently screen for pathogens while tolerating the microbes, the intestinal microbiota. Goblet cells are a key component in the regulation of the immune system. And how goblet cells are regulating the immune system. So goblet cells can form goblet cell-associated passages. Upon mucin secretion, despite the mucous fence, in response to acetylcholine acting on muscarinic acetylcholine receptor 4 in a fascinating way, goblet cells-associated passages can form, acetylcholine can form goblet cell-associated passages and deliver luminal antigens and microbial products to subjacent lamina propria dendritic cells. These dendritic cells are known to have tolerogenic properties. Here is a representation of the major cell types in the lamina propria, their major functions and the impact seen in alcohol liver disease and alcohol-associated liver disease. Here in blue, I just represented the immunocells that are more interesting now for this project. So, as I mentioned before, goblet cell-associated passages or GAPs can regulate dendritic cells, which at the same time, dendritic cells are known to induce the production of IgA, which is also important in liver disease, alcohol-associated liver disease. Also, dendritic cells are known to secrete IL-23, which in turn is going to induce IL-23 and the secretion of IL-22, which is also known to be important in the prevention against alcohol-associated liver disease. And also, dendritic cells are known to induce tolerance by inducing T-regulatory cells. Here is the representation of GAPs identified as a red rhodamine dextran filled columns transversing the epithelium and positive for MAC2 staining, shown in green. The rhodamine dextran enters into the gap, allowing us to discern them and beautifully observe the communication with the lamina propria immune cells. We observed that wild-type mice fed alirically for 10 weeks had a markedly increase in the number of goblet cells in the proximal small intestine, as indicated by an augmented area of MAC2 staining. And notably, we observed a reduced expression of the gene that codifies for muscarinic acetylcholine receptor 4, and as well as reductions of GAPs by ethanol. Therefore, we aim to modulate a GB130 pathway, a pathway that is known to have less differentiation, less number of goblet cells, and investigate whether inflammation, bacterial overgrowth and translocation, immune response, and to investigate whether these mice are protected from alcohol associated liver disease. For that, we used a genetic mouse model that expressed activated GB100 specifically in intestinal epithelial cells under the control of the Vilin promoter. This was a kind gift of Dr. Michael Karin. And we exposed these animals to a mouse model of liver urticaria for 10 weeks. Transgenic mice were protected from ethanol-induced liver disease compared with the wild-type mice, as indicated by lower levels of ALT and lower triglyceride levels, hepatic triglyceride levels. I previously mentioned that GB130 regulates the balance between proliferation and differentiation, and that these mice have lower numbers of goblet cells. This also happened, we proved that, and we also demonstrate that this was happening after ethanol consumption. Notably, mice overexpressed with active GB130 had a great increase in the numbers of gaps. The gene levels and the protein levels of the receptor muscarinic acetylcholine receptor 4 were induced also in the transgenic mice. So you can see here that the transgenic mice had increased gaps, and interestingly, it was in this area located proximal to the crypts. Among the most astonishing findings, we found that transgenic mice had increased frequencies and numbers of tolerogenic lamina-propria dendritic cells and higher numbers of IL-23 and IL-10 expressing cells. Effectively, in the lamina propria of the transgenic mice, the induced expression of IL-23 by dendritic cells induced the frequencies of ILC3s and the secretion of IL-22, which in turn stimulated the expression of antimicrobial vectors, RED3G and RED3B. And among other anti-inflammatory cytokines, we found an increase in IL-10. Concordantly, we found an increase of T-regulatory cells in the transgenic mice after ethanol. In fact, expression of active GB130, which induced the secretion of antimicrobial peptides, were not only protected from the ethanol-mediated bacterial overgrowth, but their microbiota signature was overall significantly from control mice. Gut-derived and translocated viable bacteria from sterine mesenteric lymph nodes and the liver were anaerobically cultured, and we found that the transgenic mice for GB130 were protected from bacterial translocation. Hence, these results indicate that activation of GB130 protects from ethanol-induced liver disease through stimulation of mucosal tolerance and antibacterial defense. Key takeaways. The physical barriers of the intestine are of great importance and play collective functions to maintain intestinal homeostasis, which is disrupted during alcohol abuse. Microbiota protects mice against acute alcohol-induced liver injury. Alternative therapeutic approaches that modulate the intestinal homeostasis could be used to prevent alcohol-associated liver disease. Antibiotics, fecal microbiota transplantation, probiotics, prebiotics, nutrition-based therapies, FXR and AFGF 15 and 18 therapies, as well as bacteriophages as antimitotic and antimycotics are, despite I haven't mentioned the importance of fungi in the alcohol-associated liver disease, could be also an alternative therapies. Opening small intestinal gaps to modulate intestinal homeostasis could be a promising target to ameliorate the progression of alcohol-associated liver disease. This project is funded by the Pilot Project Award from the ALPD and the Pinnacle Research Award from the ASLD. I recently also received a pilot grant from San Diego San Diego Digestive Disease Research Center, and I would like to thank all of my collaborators, especially my mentor, Dr. Schnabel, because it's been a pleasure to work with him during all of these years for his continuous support. I would like to thank all of the lab members and all of the collaborators that have contributed to all of the work presented here today, which are many. I don't have time to and I don't even put them all in one slide. Thank you all for your attention.
Thank you very much, Cristina, for this beautiful talk. It's been amazing, very beautiful data from your lab, and also your current work now. And okay, now before continuing, let me introduce you to and welcome also your mentor, Professor Snabel. Dr. Snabel, thank you very much for being here today with us.
No, in fact, in the different projects, it's been different models. So to enter into detail, for example, in the article that with the ATP4 mutant mice, we use the 2 months model of Liberty Carlis during I think it was during 8 weeks, and then for example, in the other paper, in the Nature paper, we use the 15 days plus binge model, and for example, in the what else? Yeah, basically these 2 models in this project, and then in the later project, in the gaps, and the model that I use is the chronic model. Well, actually, also in the Nature Communications, we treated the mice both. We showed that it was working in the short model and the long-term model also. And in the latest, I'm doing the long-term model. So more number of goblet cells.
In the absence of any...
I know the other way around, but oh well, yes, sorry. And actually, I've used it. Sorry, it's the AGP130 knockout mice, and actually, I haven't shown the data here today, but I did every experiment also on them, and they actually had exacerbated liver disease. So in this study, not all of the patients had cirrhosis, and also Bernd could help me more maybe with the patients, and the humans, and the disease is comparable to humans. I will say that it is comparable to humans because they develop. Because they also. Because this model is comparable to human, despite, it is still a mouse model.
Okay.
Yeah. First of all, thank you so much, Ruben and Esther, again for inviting us, and also for Christina inviting to present here. I think that's a great opportunity. The patients with alcoholic hepatitis, about 2/3 probably have underlying cirrhosis, and this was a multi-center study, so most of them actually have cirrhosis. And unfortunately, with none of the animal models with alcohol, we can achieve the same disease severity in mice. So that's a major setback for all of our studies that we do with alcohol. So that we will probably never be able to do this. To achieve this. There are a few models, especially with the intragastric feeding model that Dr. Tsukamoto developed at USC in Los Angeles. But even he, with the most severe liver disease, and probably cannot achieve the same degree of cirrhosis as we have in these very severe and sick patients of alcoholic hepatitis.
This is a very smart question, and thank you for reading our articles. I really appreciate this. So in this Nature Communication paper, when we trying to find a mechanism, and we treat we use this mouse, we found that that bone marrow transplanted mice were protected from liver injury after Enterococcus gavache. So the reason could be, and actually I've been thinking about this question many times, is that also pore-forming toxins are known to induce IL-1 beta and this pathway, the NLRP3 pathway. So probably the mechanism of death in cells, it is maybe pyroptosis. We still don't know. This is still not investigated. Probably using inhibitors such as anakinra that we also use in the model, and inhibiting these pathways is maybe preventing the death of the hepatocytes. But obviously, this is still something to explore.
Yeah, we actually have, and this is a very valid point, Ruben, that you're raising. And I already talked to the FDA, and that's also their main concern is, especially in this very, very severe sick patient population with alcoholic hepatitis, them, so can they induce like an inflammatory response, an antibody response? You have to, and I think this is always ignored, that we have, in our body, we have such a tremendous amount of phages. I mean, this is like, they outnumber the bacteria. So that's, we are a natural habitat for bacteriophages. So we all of the times we have bacteriophages in our body. So we know from the alcoholic hepatitis patients, because they have this underlying cirrhosis, they have this, their gut barrier is disrupted, like, almost non-existing any longer. So I think there's a constant influx of bacteriophages into the systemic circulation from these patients, and they're just cleared. If they don't have like a natural host, and the natural host are bacteria, right? So if there are no natural hosts there, they will essentially die. But coming back to your more question, does they elicit like an inflammatory response? And we have, up on consulting with the FDA, we have carefully looked at this, and Christina was not so much involved in this. That's why I'm answering this question. I apologize, Christina. We have not seen, in fact, like if we give phages to our mouse model, we can see them if we gavage them in this mouse model. We can see them everywhere. So we can see them in the liver, we can see them in the heart, we can see them in the spleen, but they are gone like after 24 hours. So if we give one gavage, you can detect them systemically, and they are gone like after 24 hours. But we have not seen actually a very robust inflammatory response. In fact, what we saw in the liver and also in the intestine, that they elicit like an IL-10 response. So in fact, the anti-inflammatory response. How this is mediated, we have no clue right now, but I think this is very encouraging. And like I said, I think our body is like, this great habitat for this, for a vast majority of these vast number of phages.
You mentioned a very important point, because maybe the question is the other way around, because it's been described that phages interact constantly with the immune system, with the innate immune system, and probably they're kind of cooperating in some kind of anti-inflammatory or tolerogenic response that it might help also to give resound to your results.
Yeah, I agree.
So these phages, Enterococcus faecalis phages, are very strain specific actually for faecalis. For faecalis, and also yes, there is a history. We know that with the time it could be they could develop a resistance, and so maybe I would like also Bernd to jump here, because he's working now on this, and I'm sure he has a lot to say.
Yeah, so you're completely right, Christina. So they immediately, the bacteria can immediately develop resistance. The approach we are taking is, and to overcome this resistance problem, is that we are using cocktail phages. We have done this in our paper as well. But moving forward in patients, we're also trying to use cocktail phages. We have now isolated about 50 phages that can target many strains that we have isolated from patients with alcoholic hepatitis that are cytolysine positive. And with these 50 phages, we have found now a cocktail of 3 phages that can cover essentially like 75% of the patients. So I think this is pretty good. So whatever patients we are getting, we can isolate the bacteria, we can test the bacteria if they are killed and lysed by this phage cocktail. And, if we can cover 75% of the patients, that's pretty good. And I want to point out, the beauty about this bacteriophage approach, if we eventually really can move this forward into clinical practice, is that this is, highly personalized medicine, right? So we can test every patient. Is the patient cytolysine positive? We can culture the bacteria, and then we can test each bacterium that is cultured and isolated from this bacteria. Can it be killed by the phage cocktail? And we can do this like within 24 hours. So there's a very fast turnover. And this is the future that I foresee. This is precision medicine and this personalized medicine. And I hope we can implement this, at one time. It's not easy, but so with the phage cocktail, with this personalized approach, I hope we can overcome these hurdles with the phage therapy. But thank you for this question. This is very important.
So yeah, thank you for this question. So we've seen that actually the bacterial overgrowth is very important in the proximal small intestine, as you mentioned, in the duodenum, and in my last work, I've been very focused in the proximal small intestine, basically, and the reason is because the gallbladder cell-associated passages, I haven't mentioned this, in the colon they are closed. So because they are regulated by MIDI88, and in the colon there is so much bacteria that all of the gaps are closed. Only they are open in the proximal, in the distal colon, and in the proximal small intestine the gaps are open, and then it's where it's been reported that there is an increase of the bacterial overgrowth as early as 3 weeks of alcohol abuse in mice. So it is that's why bacterial translocation is very important here, and also not only for many reasons the importance of the overgrowth in the small intestine is very important in the onset of the liver disease, not only for the immune system, bile acids and other regulators. We haven't isolated the systems. So what I mean is I don't know which ones, but we did a bone marrow chimera transplants. So a lot of immunocells are going to have this depletion in the TLR2 knockout, TLR2 and MIDI88. However, I cannot discriminate in these systems. So it will be very interesting to have. Yeah. We isolated the Kupffer cells and also the hepatocytes and the Kupffer cells, but I don't know. I don't have other in vitro cell types that I've seen it, but for sure. But it will be interesting. We will love to see that. We will really love to see that. But my belief is it's gonna take a while. But I think we already see that the use of antibiotics, it is in the future is not gonna be possible, and actually it causes so many other problems sometimes on dysbiosis and dysregulation of the immune system that we are going to have to look for alternatives like phages, and we have them here available, and we just need to investigate a little bit more about it and finally use it for the benefit and to improve and alleviate liver diseases. So I would love to see that.
Yes, sure. Doctors.
Yeah, if I can just one thing, we have, and that's also triggered our studies. We have an iPhage center at UCSD in San Diego here, and they are routinely, essentially, using phage therapy for for patients with multi-drug resistant bacteremia or sepsis. And, one of these cases was essentially the reason why this center was founded like many years ago. And this, so they essentially are identifying phages based on this multi-drug resistant bacteria. This is always like NS1, right? So they have this bacteria from one patient, they make a phage, and they give it to the phage. They usually get an emergency IND from the FDA, and then they inject intravenously into the patient's systemic circulation. In this first patient, it was actually injected. This patient had a necrotizing pancreatitis, so he had this. He developed this pseudocyst, and then they also injected the phage into this pancreatic pseudocyst, so which is not a pseudocyst. This was a real cyst, and so we have, the application is there. We just need to get this, hopefully overcome many of these problems like we have discussed before with the resistance, with the safety and so on. We have to overcome this, but I think it's not a short, it will take us a while to do this, but hopefully we get there at one point.
And I think that one possibility for, going further in this personalized medicine maybe would be whenever we are ready to find a way to deliver to specific locations, whatever, which is the treatment that we want to use, in this case, these phages. I think that at the moment that we can find that technology that we can deliver those phages to specific locations in the gut, I think everything will go faster and will be closer to treat patients.
Yes, I agree.
Thank you, everybody. Thank you all for being here today.
Thank you all.
Thank you.